morning, I am Madhurima Datta, Zoology and Biology educational content creator. Now content for B.Sc and M.Sc in Zoology honors, Physiology honors, Microbiology honors, Botany honors and Biotechnology honors. An exam for CUT PG exam, GATE exam, DBT exam, IIT JAM exam and CSI NET Life Sciences exams. Paper Immunology, Chapter 2, Cells and Tissue of the Immunosystems. And today I will discuss about the monocytes and macrophages. What is the mononuclear phagocytes or monocytes? The mononuclear phagocyte system includes circulating bone marrow derived cells called monocytes. So, many of which become macrophages when they migrate into tissue, and tissue resident macrophages which are initially derived from yolk sac or hematopoietic precursor during fetal life. So, monocyte is derived from the bone marrow. It is an immunocyte which is circulating in the blood with a short lifespan of 1 to 7 days. And macrophages are mature cells which is formed when circulating monocytes migrate into the body tissue. Next, development of macrophages and monocytes. First, mature cells formed when circulating monocytes migrate into the body tissue. First pathway is bone marrow pathway. It is called bone marrow pathway because after birth, cells of the monocyte macrophages lineages arises from committed precursor cells in the bone marrow driven by a cytokine called monocyte or macrophages. Its colony stimulation factor, also called MCSF. And this precursor matures into monocyte which enter the circulate in the blood where they have a short lifespan of approximately 1 to 7 days. Next pathway is also called fetal pathway because blood monocytes are efficiently recruited. Most long-lived tissue resident macrophages are derived not from the bone marrow but from yolk sac or fetal liver precursors during the fetal development. These cells have self-renewal capacity, so they can maintain stable numbers. Next pathway is specialized tissue and this pathway is also called self-renewal pathway. And the specialized tissue are Kupffer cells and alveolar macrophages in lung and microglial cells in the brain. So, they often assume specialized phenotypes depending on the organs. Example are Kupffer cells lining the sinusoids in the liver, alveolar macrophages in the lung and microglial cells in the brain. Next is monocyte. Introduction of the monocyte. In the steady state, blood monocytes are recruited at a low rate into the healthy tissue. And why they differentiate into tissue resident macrophages? This pathway of monocyte differentiation into tissue macrophages supplements the self-renewal of the fetally derived cells and accounts or vary in reactions of resident macrophages in different tissue. Next, characteristics of the monocytes. Here I will discuss about the size and structure, subset and surface markers of the monocyte. First, size and structure. Monocytes are 10 to 15 micrometer in diameter and they have bean-shaped nuclei and finally granular cytoplasm which is containing lysosome and phagocytic vacuoles and cytoskeletal filaments. Next, subset of the monocytes. All human monocytes expresses class 2 major histocompatibility complex, that is M.Sc molecules, and CD11b and CD86, and all mouse monocytes express CD1115 and CD11b and CD64. This is the surface marker of the monocytes. However, monocytes are heterogeneous and consist of different subsets distinguishable by cell surface markers and function but not morphology. In both human and mice, the most numerous monocytes called classical or inflammatory monocytes, which is produce inflammatory mediators, are phagocytic and are rapidly recruited to sites of infection or tissue injury. Next, non-classical monocytes. The second type of circulating monocytes called non-classical monocytes. It is a 5% to 2% of blood monocytes which are recruited into tissue after infection or injury and may contribute to repair. Next, classical or inflammatory monocytes are most often distinguished from non-classical monocytes by their relatively high expression of CD14 or LY6C and CCR2 mice. Some non-classical monocytes are known to crawl along endothelial surfaces where they scavenge luminal microparticles and may play a role in elimination, circulation, microbes and in repairing endothelial barrier defects. The developmental relationship between monocyte subset is not fully understood. Next, functions of the macrophages. First is ingestion. A major function of monocyte-derived macrophages is in host defense is to ingest microbes by the process of phagocytosis and then to kill the ingested microbes. Next, amplification. Tissue-resident macrophages function as sentinel cells that sense the presence of microbes and respond by secretion cytokines that initiate and then amplify the protective response against the microbes. Next function is pyroptosis. Macrophages that have engulfed microbes can be induced by microbial molecules to under of an inflammatory form of death called pyroptosis, which usually result from activation of a cytoplasmic enzyme complex called the inflammasome. Next, role in apoptosis. In the addition to ingesting microbes, macrophages ingest necrotic host cells, including cells that die in tissue because of the effect of toxins, trauma, or interrupted blood supply and neutrophils, and that die after accumulation at sites of infection. This part of the cleaning up process after infection or sterile tissue injury. Macrophages can specifically recognize and engulf cells that die by apoptosis before the dead cells can relate their content and induce inflammatory response. And last function is antigen presenting cells. Macrophages roles and as a antigen presenting cells or APCs. That is, macrophages serve as antigen presenting cells that display fragments of protein antigens to T lymphocyte and activate T cells recruited to sites of injury or infections. This function is important in the effector phase of T cells mediated immunoresponse. Macrophages promote the repair of damaged tissue by stimulation new blood vessels growth, that is called angiogenesis, and synthesis of collagen-rich extracellular matrix, that is also called fibrosis. These functions are mediated by cytokines secreted by the macrophages that act on various tissue cells. Now the topic has ended. For all video of B.Sc and M.Sc in Life Science and Biotechnology, please subscribe my YouTube channel Biogenesis by Madhurima and follow my Facebook account Madhurima Datta. Thank you everyone.